Hi students, in this video lecture I would like to explain you about drugs acting on genetic material. As you already know about drug is a chemical substance. Drug is a chemical substance which interacts with macromolecular targets, macromolecular targets in the body and produces pharmacological effect. Okay, so this is the definition for drug. Just I would like to remind you the definition and we'll move forward. Okay, it is a chemical substance and it interacts with the macromolecular targets in the body and produces pharmacological effect. Okay, so that is about drug. Okay, so drugs acting on genetic material. So let me introduce you about genetic material. What is genetic material? Genetic material is nothing but DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Okay, so these two materials can be called genetic material. Okay, deoxyribonucleic acid and ribonucleic acid. And let me give you some clear details about this genetic material. First, let me explain you about DNA deoxyribonucleic acid. Deoxyribonucleic acid. Okay, DNA. Okay, because genetic material consists DNA and RNA in their structure. Okay, so in deoxyribonucleic acid, so that means every cell contains the nucleus and has a genetic material like this. Okay, so this is the double helix of DNA. DNA contains two strands and which are come together or attached together this chain with the hydrogen bonds in between base pairs. Okay, so in between this chain forms with the bases. What are the bases in a DNA chains? Okay, so adenine and guanine. These two bases are called purines. And thymine and cytosine, these two bases together called pyrimidines. Okay, pyrimidines. Okay, so in double helix of DNA, the chemical structure is composed with the bases. What are the bases? Adenine and guanine. Both of them are together called purines, and thymine and cytosine, they are called pyrimidines. Okay, so these are the bases exist in DNA or RNA molecules, right? So in in RNA extra uracil, okay, another base which is only present in RNA, it will not present in DNA, double helix structure of DNA, okay. So double helix structure of DNA is like this, okay. The two chains attached together with this coil like structure, helical structure, and in between, so it has bases like adenine. So another chain thymine, in between there is a two hydrogen bonds, okay. And in the case guanine and cytosine, in between there are three hydrogen bonds which keep them together. Because of these interactions, double helix of DNA stay together like this helical chain, okay. So in between it has bases like adenine and guanine, both of them together called purines, and thymine and cytosine, both of them called pyrimidines. So in the case of RNA, uracil is present, but whereas in the case of DNA, deoxyribonucleic acid, so uracil will not be there, okay. So here these bases, these single strand DNA, okay, two strands of DNA, this is one strand, okay, one strand of DNA, and this is another strand of DNA, another strand of DNA, okay, double helix DNA, okay. So these chains composed with the bases, okay. What are the bases? Adenine and guanine and thymine and cytosine. So in between, that means adenine always forms hydrogen bonds with the thymine ATGC with the two hydrogen bonds, and guanine always forms three hydrogen bonds in between adenine, guanine and cytosine, okay. So here two hydrogen bonds, these are the two hydrogen bonds. What is hydrogen bond? You know, here three hydrogen bonds in between, hydrogen bonds, okay. So hydrogen bond is nothing but the interaction between the hydrogen which is attached to more electronegative atom and more electronegative atom, okay. So the bond formation of the interaction between the most electronegative atom and the hydrogen which is attached to the most electronegative atom is called hydrogen bond, okay. So these helical structure of DNA, double helix of DNA contains like these bases adenine and guanine and thymine and cytosine, okay. And adenine always form hydrogen bonds with thymine, and guanine always forms bonds with this cytosine ATGC, like so they are base pairs like that, okay. So just let me give you some the structures of adenine and guanine, okay. Adenine and guanine like this. So this is the structure of adenine. Okay, adenine. Okay, so this is one of the base in the DNA structure, and the guanine structure would be like this, guanine. Okay, guanine, structure of guanine. So this is the guanine structure, guanine. So these are the two bases which can be considered as purines. And about pyrimidines, okay, these are these two can together consider as purines, purine bases. Okay, and about adenine, about thymine and cytosine. So this is about sorry, here it is double bond O. Okay, hydrogen. So this is about cytosine and so okay, this is cytosine and this is thymine. Okay, so both can be together considered as pyrimidines. Okay, these are pyrimidines. Okay, so these two bases are considered purines and these two bases considered pyrimidines. Okay, so these are the bases exist in DNA structure, and along with this bases there are also sugar moieties are present. So what are the sugars exist in DNA? What is the sugar structure exist in DNA? So this is the sugar structure. Okay, this is, what about this? Hydrogen and OH. So this is deoxyribose. So this is the sugar moiety exist in DNA helical structure. Okay, so here if you give the numbering 1 2 3 4, okay, and 5, so 2 deoxyribose at second position, hydroxyl group, okay, oxygen will be removed, therefore 2 deoxyribose. Okay, what is the ribose structure? Ribose structure is this one. Okay, a slight difference between ribose and 2 deoxyribose, OH and hydrogen, OH, hydrogen, OH and hydrogen, H and OH. So this is the ribose structure. Okay, So this is ribose structure, and this is 2-deoxyribose. That means at second position this oxygen will be removed in the case of DNA. Therefore 2-deoxyribose nucleic acid. Okay, RNA, in RNA this ribose sugar present. Okay, so that is the difference in between DNA and RNA. DNA is double helix helix structure it has, and in the case of ribose or RNA it has single strand structure. Okay, so these are the chemical structure present in this molecule, in this materials, in these genetic materials. Okay, so three materials. Okay, another material is phosphate. Okay, phosphate is like this. Okay, so this phosphate also exists in DNA structure or RNA structure. Okay, so overall I want to tell you that in DNA and RNA what are the chemicals present is, one is bases. Bases means adenine, guanine, thymine and cytosine. In the case of RNA, uracil also. Okay, and sugar moiety. Okay, and phosphate groups. Okay, so these are the chemical moieties present in DNA and RNA structures. Okay, double helical structure. Okay, if you would like to understand the structures, chemical structures of DNA, so what is the genetic DNA structure is double helical structure like this. Okay, like this. So each chain composed with bases and sugar moiety and phosphate. Okay, so one end is from 3' to another 5'. It is opposite ends, 3' and 5'. Okay, so these are the numbering where one nucleotide attached to the another nucleotide. Okay, so DNA or deoxyribonucleic acid, the structure of DNA, it has double helix structure. It is in helical chain, right? Okay, and each chain composed with bases, sugar moiety and phosphate. Okay, and both the chains stay together, and they can be in compressed mode due to the interactions between them. The bases is hydrogen bonding. Okay, so these are the details about DNA chemical structure. Okay, so this is the deoxyribose and ribose and these bases, and this is phosphate group. Okay, I'll give you one example structure for DNA. Okay, generally just I'll write the structure. Okay, so this is the guanine structure, how it connect with the sugar moiety, Okay, so this is the like this, hydrogen and hydrogen, OH, hydrogen, O, and phosphate group. So this is the nucleotide. Okay, so the base, this base attached to the sugar moiety in this way, and this sugar moiety attached to this phosphate. Okay, so three of them together called nucleotides. Okay, so when this base and sugar moiety forms together, they can be considered as nucleoside, but three of them, this can be called as nucleotides. Okay, nucleotide. In nucleotide we have base and deoxyribose sugar and the phosphate. Okay, three of them together called nucleotide. Like this, it forms bonds with another moieties and forms so at this is the 3' position. Okay, and this is the 5' position. Okay, and here, so another nucleotide will be attached at this junction, at this position. Okay, so this is the way DNA molecule composed. Okay, so I hope you understand about DNA structure. Okay, in DNA structure, it has simply you can remember that it has bases like adenine and guanine, and cytosine and thymine. Okay, and sugar, deoxyribose sugar, and phosphate moiety. Okay, so these are the simple bases and sugars and phosphates together, combined together, and forms DNA double helical strand, and in between there are hydrogen bonds. Adenine always forms bonds with thymine with 2 hydrogen bonds. Guanine always forms 3 hydrogen bonds with cytosine. So these are the base pairs. Okay, in between. So in between means, so here double helical structure of DNA is like this. Okay, so here one strand A, another strand C, that means cytosine T. So in between there is 2 hydrogen bonds like this. So double 2 strands come together and stay together because of the interaction, hydrogen bonding interaction in between them. Okay, and this is the double helix of DNA, helical structure. Okay, so in DNA helical structure, double helical structure, so there are minor groove binders, minor grooves, and also they have major grooves. DNA helical structure have minor grooves as well as major grooves. Okay, depends on the size of the groove. Okay, if it is smaller, then minor groove; if it is larger, then major groove. Okay, so the drugs which are able to insert in between DNA chains through these minor grooves and major grooves, you should remember this point. Okay, the drugs which synthesized as the drugs acting on genetic material, that means the drugs, these kind of drugs slip into DNA double helical structure and interact with the DNA through minor grooves or major grooves. Okay, so this you need to remember. So this is about the structure of genetic material. Okay, the structure of genetic material, the structure of genetic material. Okay, so you know about drug, the definition of drug, and also genetic material. Okay, in this unit you are going to learn the interactions. You are going to learn the interactions between drug and the genetic material, and how it alter the replication or translation, okay, transcription processes. Okay, how they can be helpful in treating some kind of diseases. Okay, so drugs acting on genetic material may be anti-cancer agents, anti-cancer agents, anti-cancer agents, antimicrobial agents, antimicrobial agents. They can be act as anti-malarial agents. Okay, anti-malarial agents. Okay, anti-cancer agents. Examples for anti-cancer agents: Adriamycin. Okay, Adriamycin or danomycin. So these are the natural products which can act as anti-cancer agents. Okay, and antimicrobial agents. Antimicrobial agents are like ecridins. Okay, ecridins: amsacrin, nitrocrine, quinocrine. Okay, like amsacrin, nitrocrine, quinocrine. Okay, quinocrine. So these are the examples for antimicrobial agents. So what about anti-malarial agents? Chloroquine, right? Chloroquine. So this is the example for anti-malarial agent. So these drugs, the example drugs, are acting on genetic material, and therefore they can be considered as anti-cancer agents. Anti-cancer agents, that means adriamycin and danomycin can act as anti-cancer agent through acting on genetic material. And antimicrobial agents, examples amsacrin, nitrocrine and quinacrine. So they can act on genetic material, and they will work as antimicrobial agents. And anti-malarial agents like chloroquine, it is also act on genetic material and act as anti-malarial agent. Okay, so these are the examples for drugs acting on genetic material. I am going to explain you one by one about them and their structure. Okay, and the mechanism of action also I am going to explain for these drugs. Okay, first let me give you example. Generally, drugs acting on genetic material, it should have planar structure, planar aromatic or heterocyclic or ring structures. Okay, rings they should have. Okay, and why it should be planar, why I mentioned specifically about planar. Okay, they should be planar, because if these kind of drugs wants to insert or incorporate into in between, okay, planar structure needed for aromatic or heterocyclic rings, because these kind of drugs should incorporate or insert in between two double helix strands. Okay, two strands of DNA. It should enter through DNA minor groove binders or DNA major groove. Okay, and it should slip into those two strands, to slip or to enter into the two strands. Between the two strands it should have planar structure. That's why I am telling planar aromatic or heterocyclic rings they must have. Then they can be able to slip into in between the two strands of DNA and forms interactions with the DNA molecule. So finally it exhibit some anti-cancer activity in that. Okay, so generally anthracyclines 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 are naturally occurring antibiotics. Okay, antibiotics. Okay, and these anthracyclines can be are isolated from Streptomyces species. Okay, so these can be isolated from these Streptomyces species. Okay, and anthracyclines are naturally occurring antibiotics, and they can act as anti-cancer agents and anti-malarial or antimicrobial agents. Okay, so anthracycline structures, I'll explain you about the structures, right? So it should be planar. That's what I am telling you. It should be planar. Okay, the aromatic rings should be planar. Tetracyclines. Okay, and this is double bond O, and here OME, and here hydroxyl group and hydroxyl group, and here CH2 R prime. This is another hydroxyl group. No double bonds in between. Okay, and here another hydroxyl group, hydrogen and hydroxyl group. Hydrogen and hydrogen, NH3 plus, hydrogen, R2 R3, CH3, hydrogen. So this is the structure of tetracyclines. Okay, anthracyclines or tetracyclines. Okay, this is the structure of anthracyclines. Okay, this tetracyclic structure. Okay, so if R prime, R2 and R3, that gives different if R prime is OH, and hydrogen and OH, so this is called Adriamycin. Okay, Adriamycin structure. Okay, and whereas if R prime is equal to H and OH, So that is called danomycin. Danomycin. Okay, so these are the anthracycline structures. Okay, anthracycline structure. And if here hydroxyl group and hydrogen hydroxyl group, then it is called adriamycin, the best anti-cancer agent ever discovered. Okay, the best anti-cancer agent, anti-cancer natural product, okay, is adriamycin. Adriamycin. Okay, so here it is hydroxyl group and hydrogen OH. So that is the structure of adriamycin. And what is the difference between adriamycin and danomycin? In danomycin, this R prime is equal to H and R2 is equal to H and R3 is equal to OH. Okay, so the slight difference between adriamycin and danomycin. Okay, so they are natural products, naturally isolated from anthracyclines as streptomyces species, and they can act as anti-cancer agents. Okay, naturally occurring or isolated anti-cancer agents. So example is adriamycin or danomycin. Okay, so you have to remember these interesting things. Okay, these can be act as anti-cancer agents. So that means this structure, this planar structure, okay, rings are in same plane. This planar aromatic or heteroaromatic rings are able to slip or able to insert in between the two chains of DNA double helix. And when it insert properly inside in between the two double helix of DNA, they can interact with the bases or DNA, okay, through different interactions. What kind of interactions takes place between the DNA and anthracyclines? Interactions are like this: hydrogen bonding interactions, and ionic interactions, and hydrophobic interactions, and van der Waal interactions, okay. So these are the kinds of interactions takes place in between drug and DNA. Hydrogen bonding interactions, ionic interactions, how it can be able to produce ionic interactions, because here you have NH3 plus. Okay, this ion can interact with the phosphate ion in the DNA chain, because DNA chain has phosphate ion. Okay, so therefore this NH3 plus can interact with the phosphate minus ion. So that kind of ionic interactions takes place. And hydrogen bonding, that means this hydroxyl group in adriamycin can form bond with guanine, hydrogen bonding with the guanine keto functional group. Okay, and hydrophobic interactions means these aromatic rings with the bases, there would be hydrophobic interactions in between them, and van der Waal interactions also takes place in between drug and DNA molecules. Okay, so these are the kinds of interactions takes place, and because of those interactions, these drugs forms stable complexes, stable complexes with DNA. Okay, and topoisomerase 2. Topoisomerase 2. This is one of the enzyme which helps in replication process. Topoisomerase 2. Okay, so during the replication, DNA and topoisomerase 2 complex will be formed, and these drugs, anti-cancer drugs, Adriamycin or Danomycin, insert in between the double helix of DNA and stable this complex for long time. Actually, normally what happens during the replication, DNA and topoisomerase 2 complex will be formed for short time, and after the completion of that work, both can be divided and divides and have their original states. But in this case, what happened, these anti-cancer agents stabilize this complex for a long time. What complex? DNA and topoisomerase 2 complex. Okay, normally DNA topoisomerase 2 complex forms and goes their original state after the completion of the process. But when these drugs treated that patients, what happens, these drugs enter into the DNA double helix in between them, insert and stabilize this complex for long time. Therefore it will not be breakable, so that the DNA distortion occurs. Okay, because of this DNA distortion occurs. So that means replication stops. Okay, replication or transcription or translation processes or translation processes will be stopped and okay, DNA distortion occurs. Because of that, they can be act as anti-cancer agents. Okay, dear students, I hope you understand the information which I provide you in this class. Okay, simply you should remember these things. Okay, so this is the mechanism of action, how anti-cancer agents work to kill the DNA. Okay, to stop the DNA replication or transcription and translation processes, due to stabilize the complex of DNA topoisomerase 2 complex. Okay, these drugs stabilize the DNA and topoisomerase complex in DNA double helix strands for long time. Because of that, those processes will be inhibited and the cells will be destroyed, the infected cells will be destroyed. That's how these drugs can be act as anti-cancer agents by acting on genetic material like DNA. Okay, so these are the things you need to remember, and I will continue in the next class about remaining things. Okay, before that I'll also explain you about some kinds of classification. Okay, drugs acting on DNA the genetic material can be classified into several types. Okay, drugs, intercalating agents. Okay, intercalating agents, okay, DNA intercalating agents, and DNA binding and nicking agents. So that means these kind of drugs bind with the DNA and cut the DNA chains. So they can be classified as DNA binding and nicking agents. And drugs, DNA polymerase inhibitors, DNA polymerase inhibitors. Okay, DNA polymerase inhibitors. Fourth one is RNA polymerase inhibitors. Fifth one is drugs interfering with translation processes. Okay, so these are the 5 kinds of drugs which are acting on genetic material. Okay, DNA intercalating agents. So just now I explained you about DNA intercalating agents. Intercalation means the process of the drug insert in between the process of. So let me give you the explanation of DNA intercalating agents. Okay, the drugs slip into or in between the drugs slip in between DNA double helix and forms interactions and distort the DNA is known as DNA intercalating agents. Okay, so DNA intercalating agents means the drugs which are slip into the double helix of DNA and forms interactions with the DNA and distort the DNA structure. That means it stops the replication processes. So because of that, there will not be any cell growth and the cell will die. So that is the effect of DNA intercalating agents. Okay, so this process is called intercalation. Intercalation means the drug insert in between the DNA double helix is known as intercalation. Okay, these kind of agents, anti-cancer agents, I gave you already examples like Adriamycin and Danomycin and acridins type of molecules like amsacrin, nitroquine and quinacrine. Okay, and next DNA binding and nicking agents and DNA polymerase inhibitors. That means these the drugs which are inhibit the DNA polymerase enzyme and RNA polymerase inhibitors. That means the drugs which are inhibit the RNA polymerase enzyme activity. Okay, and I'll give I'll give example for each in the next classes. And the finally drugs intercalating with translation process or transcription processes. Okay, so these are the 5 kinds of classification are there the drugs acting on genetic material. Okay, so in this class I explained you about DNA intercalating agents. Okay, example is Adriamycin and Danomycin. Okay, they intercalate in between. Okay, and also I'll explain you in the next class about antimicrobial agents like amsacrin, amsacrin and nitrogrin and quinacrine. And also I'll explain you about chloroquine, which is act as anti-malarial agent. Okay, so if you have any doubts in this class or in this lecture, so you can post your doubt in the comment box so that I can answer to those doubts, and I can clarify those doubts for you. And if you like this class, you can click on the like button. And also, if you listen my class first time, or if you are new to this channel, you can subscribe to this channel. Okay, thank you, bye.